Welcome everyone, my name is Sebastian. Today I would like to talk to you about our work on scalable axiomatic explanations of deep neural networks for Alzheimer's diagnosis. In this work, our focus is on deep learning for Alzheimer's diagnosis. We consider networks that expect heterogeneous data as input, namely a combination of tabular biomarkers and brain morphology. For brain morphology, we consider a point cloud representation of the hippocampus because of its important role in Alzheimer's. The network is trained to predict the probability of dementia from the 3D hippocampus shape and tabular biomarkers. We denote such a network by the function f and will assume that it has been trained successfully and achieves high predictive performance. While high predictive performance is a first step towards deploying such a model, we still have the issue that its decisions are opaque, which means we require post-hoc techniques to explain those decisions to a user. Therefore, our work falls under the broad category of explainable AI. In particular, we seek to explain to the user how the prediction for one specific input came to be. To this end, we assign each part of the input a score that corresponds to its marginal contribution to the predicted probability of dementia. There are three aspects that make explaining an Alzheimer's diagnosis challenging. First, we have to consider multiple types of inputs, namely the 3D anatomical shape and tabular biomarkers. Second, in contrast to an image, a point cloud is a non-Euclidean representation. And third, to learn from anatomical shape, we have to employ architectures that differ substantially from DNNs, which most previous work in explainable AI focused on. Next, let's have a look at some existing explainable AI methods, which are summarized in the table shown here. The rows are the different methods and the columns denote the fundamental axioms that have been established in previous work. The important part here is that the Shapley value in the last row is the only procedure that satisfies all of these axioms. These theoretical results motivated us to provide an explanation by estimating the Shapley value of all parts of the input. At the top, you have the definition of the Shapley value. The Shapley value of feature I of the input set is its average contribution across all coalitions with other features. Therefore, it considers all possible subsets S of the full set of features F. The difference marked in blue compares the prediction of a model that has access to feature I with the prediction of a model that does not have access to feature I. To avoid retraining for each subset, we follow the common approach in the machine learning literature that simulates the absence of a feature by replacing its value with the value of a baseline or reference input instead. For images, the baseline is often a black image, but since we are dealing with shape representation, we instead project each point onto the convex hull. The downside of the Shapley value is that it scales exponentially in the number of features, which means we need to approximate it for most practical purposes. The figure at the top shows the architecture of the network we are using to predict dementia. This network has been proposed in our previous work and is essentially a combination of a point net that takes a point cloud and a linear model that takes the tabular data. Therefore, it is called a wide and deep network. This model has the advantage that we can estimate the Shapley value of the tabular features by just looking at the coefficients of the final linear layer. This is because a linear model has no interactions among features. Unfortunately, when estimating the Shapley value of a point of the hippocampus shape, we have to consider the entire point net, which scales exponentially in the size of the point cloud. In this work, we propose a method to approximate the Shapley value efficiently. In the end, we will lower the runtime complexity from exponential to quadratic using a combination of approximations. First, we reformulate the Shapley value by splitting the sum into two sums such that the inner sum is over subsets of equal size. Then, the inner sum can be approximated by an expectation over sets S of size k. In the remainder of this talk, I will focus on estimating this expectation using only two forward passes. Due to the linearity of the expectation, we can split it into two parts. One that considers feature I and one that does not. Therefore, we will use one forward pass to estimate each of these expectations. To reiterate, we need to estimate the expected prediction of the point net where k points have been replaced with points from a baseline shape. Our proposed solution has two main steps. First, we approximate the output of the first fully connected layer of the point net by a normal distribution. This in turn enables us to treat estimation of the Shapley value as a problem of propagating aleatoric uncertainty. The trick is that propagating aleatoric uncertainty is exactly what lightweight probabilistic deep networks proposed by Gust and Roth already do. Let's focus on the normal approximation of the first layer. The first layer is fully connected, hence it computes a sum over products as shown in the formula for Hj. But the expectations above is with respect to the network's output where k points have been removed. We don't know which ones have been removed, only that k have been removed. Our objective is to determine the mean and variance of the outputs of the first layer. The clue is that a fully connected layer computes a sum, and sampling theory tells us that the sum over a random subpopulation of fixed size can be approximated with a normal distribution with the mean and variance as shown at the bottom. Now we completed the first step, that is, we approximated each output of the first layer by a normal distribution. Next, we need to propagate these distributions through the point net, for which we transform the point net into a lightweight probabilistic network. This is relatively easy, we just need to replace each layer by its probabilistic counterpart. This leads to the main result of our paper that allows us to estimate the expectation over sets of size k with a single forward pass through the lightweight probabilistic network. Therefore, one Shapley value can be computed in linear time and all Shapley values in quadratic time. In our paper, we performed two experiments. The first experiment uses synthetic data to quantitatively compare our approximation with the exact Shapley value, but due to time constraints, I will skip this part. Instead, I will focus on our second experiment on real data from the Alzheimer's disease neuroimaging initiative. In this experiment, we use a wide and deep network architecture and train it for control versus dementia classification. For each patient, we extract a point cloud of the hippocampus and use 9 tabular biomarkers. The model performs very well with a balanced accuracy of 0.942 on the test data. The first plot provides an overall view on all 167 correctly classified patients of the test data. For each feature, 1 point represents the Shapley value of one particular patient. The features are sorted by average importance, therefore hippocampus shape is on average the most important, followed by FDG PET. The colors of the points indicate whether a feature has a high or low value. For instance, if we look at AV45 PET, it tells us that the model associated high values, indicated in red, has stronger evidence for dementia than low values, indicated in blue. This is in line with our medical understanding that a higher amyloid load in the brain is associated with cognitive decline. The second plot shows the Shapley values for one particular patient. This provides us a detailed explanation about the contribution of individual parts to the model's prediction. For this patient, the hippocampus shape is the most important feature, followed by the tabular biomarker p-tau. The third most important feature is FDG PET. The arrow to the left and its turquoise color indicate that the model thinks that FDG PET is evidence for diagnosing the patient as healthy. Overall, the results are reassuring because the model utilized the data in a way that agrees with known medical knowledge on Alzheimer's disease. The previous plot summarized the contribution of the entire hippocampus structure as a single Shapley value. This plot shows the Shapley values of individual points of the hippocampus point cloud. It reveals that the most important points, colored in red, are located in the CA1 subfield of the hippocampus. This is another reassuring explanation because atrophy in the CA1 subfield is an established marker for Alzheimer's disease. To summarize, our proposed approach is based on the Shapley value, which is the unique procedure that satisfies all desirable axioms for local explanations. We approximated the Shapley value by using results from sampling theory and propagation of aleatoric uncertainty to lower the runtime complexity from exponential to quadratic. Finally, we have demonstrated the practical value of our approach to explain Alzheimer's diagnosis by a deep neural network from anatomical shape and tabular biomarkers.